Welcome to our BioLegends scientific poster video review. Today, we'll be focusing on microglia, the resident macrophages of the brain and spinal cord. Dysregulation of these cells can lead to neuropsychiatric and neurodegenerative disorders. So, it's important to have the right research tools to identify these cells and distinguish them from other myeloid cells in the central nervous system, or CNS. Murine purinergic receptor P2RY12, also known as P2RY12, is a G protein coupled receptor for ADP and ATP. It was originally discovered on platelets, but has since been identified on microglia. Our new clone S16007D, which we'll be referring to as S16, specifically stains mouse P2RY12 and does not react with human samples. This clone helps to identify microglia and is compatible with flow cytometry, immunohistochemistry, and cell separation assays. First, we check to make sure clone S16 properly recognizes its target. We used a P2RY12 transfected cell line to overexpress mouse P2RY12 and GFP. Compared to the isotype control data on the left, clone S16 stains 99% of the cells in the sample on the right. Next, we wanted to confirm that P2RY12 was co-expressed with other microglia markers like CX3CR1 and IBA1. In these images, we stained formalin-fixed paraffin-embedded mouse brain samples. P2RY12 staining with clone S16 is shown in red, CX3CR1 in green, and IBA1 in blue. As you can see, all three markers are co-localized in this immunohistochemistry staining. Now that we've shown P2RY12 can be found on microglia, we wanted to be certain that this was specific to only microglia in the CNS, and not on other myeloid cells found in the periphery. By flow cytometry staining, we tested several other organs, including liver, lung, and spleen. Only the brain sample showed strong co-staining for P2RY12 and CX3CR1. We also wanted to examine the change in microglia marker expression upon inflammation. Lipopolysaccharide, or LPS, is an endotoxin that promotes secretion of pro-inflammatory molecules when administered to mice. Therefore, it is a potent inducer of inflammation. For a low-dose LPS treatment, we injected 25 micrograms of LPS into mice once a day for 4 days. The high-dose treatment involved injecting 50 micrograms of LPS once a day for 2 days. Daily PBS injections over the course of 4 days served as the negative control. Single-cell suspensions from mouse brain were generated and stained for the indicated markers. As you can see, CD11B expression increases with exposure to LPS, indicating the microglia are likely becoming activated and promoting neuroinflammation. Conversely, P2RY12 expression decreases upon exposure to LPS, in agreement with previous publications that indicate P2RY12 can become downregulated under inflammatory conditions. Our MojoSort magnetic cell separation system is a helpful tool for pulling cells of interest from a heterogeneous population. This can be particularly important for researchers trying to isolate microglia from the brain. The MojoSort mouse P2RY12 selection kit labels microglia with a biotinylated anti-P2RY12 antibody. Then, we use streptavidin nanobeads to bind the cells of interest and adhere to the applied magnet. Gating on CD11B and CX3CR1 double positive events, you can see that our kit isolates microglia at nearly 100% purity from a single-cell suspension of brain tissue. In conclusion, we've generated a new mouse-specific antibody for P2RY12 detection. Clone S16 is useful for several applications, including immunohistochemistry, flow cytometry, and cell isolation. And for convenience and a simplified workflow, direct conjugates of clone S16, like PE and APC, are available for use. We've shown that this antibody recognizes its target effectively in both in vitro and in vivo scenarios, allowing researchers to confidently and effectively identify microglia in their samples. You can learn more about microglia and view the scientific poster based on clone S16 in the links below. Thanks for watching our video. We hope it's been informative and that you have a new appreciation for microglia markers. Stay tuned for more videos detailing our scientific studies in the future.